Good morning, everybody. We are a little bit already over time. Let me greet everyone who's awake at 8 o'clock on Saturday. And the hero of the day, the first speaker, who's our distinguished invited speaker, David Everman. He will show us the mysterious world of split hand and foot malformations. Welcome, please.
Thank you. All right, well, I certainly want to thank the organizers for the kind invitation to come to the meeting and the opportunity to speak. And I do want to mention that I don't have any disclosures to make. So today I want to talk about split hand foot malformation, which is abbreviated throughout the talk as SHFM. And this is a very specific congenital limb malformation that has been of great interest at the Greenwood Genetics Center for many years, and I've certainly had a great interest in this condition for a long time. And so I hope that I'll be able to illustrate that although this condition looks fairly straightforward on the surface when you just look at the hands and feet of a given individual, that in fact there are many layers of complexity to it that are still being unraveled at the genetic level and the molecular level by many people throughout the world. So I'd like to start with this photograph from the 1800s. So this was taken by a famous photographer of the time named Charles Eisenman, who was interested in photographing individuals with physical differences. And this illustrates a couple of important points about this condition. So first of all, it sort of shows you the very typical or classic appearance of the hands. So typically you see absence or underdevelopment of the second, third, or fourth digits in the hands or the feet. So you see this central deficiency of the hands and feet. This often goes down into the level of the metacarpal or the metatarsal bones, and so you see these deep clefts that give the hands this very characteristic split appearance that's been called various things over the years. And you can see from the title of this photograph, Lobster Boy, that this was a term that was used for many years and in fact is still used. So if you actually look in PubMed, you'll find very recent publications that still use the term lobster claw deformity for this malformation. But certainly this is a pejorative term that we would greatly discourage anyone from using with patients and families. It's not really an appropriate term to use, but it is still out there. You will encounter that. Another term that is also not really the most specific term to use, but is also very commonly used, is ectrodactyly, and that literally means abortion of the finger. And the term ectrodactyly really means digital absence of a variety of types and patterns, but many people over the years have used that term as a synonym for split hand foot malformation. So you will see the word ectrodactyly in a number of the syndromes that include split hand, split foot malformation. And so I think this particular malformation obviously is just one larger piece of one smaller piece of the larger group of congenital limb malformations. And so I wanted to talk a little bit more about congenital limb malformations as a whole and then come back to split hand foot malformation to use it to illustrate a number of important themes that have sort of become evident about congenital limb malformations as a whole. So limb malformations are relatively common. They're easily visible, so they're fairly easy to diagnose, but some people do hide their hands so that they're harder to see, or if you don't take off the shoes and socks, you may not see them. They obviously have a significant impact on patients' lives, both on their function and also on their appearance and their feeling of well-being. But that being said, many people, including young children, adapt to limb malformations very, very well. They are often genetic, but not always genetic in origin. But because they're often genetic, they certainly have a significant chance to recur in families, and so that's often a very significant concern that parents have is the chance of recurrence, which can sometimes be difficult to really sort out. And of course, they may be part of a larger condition, an underlying syndrome with other important manifestations that we need to be aware of. And so when we see patients who have limb malformations or limb anomalies of various kinds, there are a number of different goals that we have in terms of trying to understand what's going on and answer the relevant questions. So we want to try to understand the extent of limb involvement, the specific pattern of limb involvement, because patterning can be a very important clue to the genetic cause in particular. We want to understand, is it an isolated or a syndromal condition, and regardless of which type it is, the limb malformation itself can obviously have important functional and cosmetic implications, but if it is part of a larger syndrome, there can be other potential medical or developmental problems that we want to be aware of. We focus a lot on trying to figure out if the things that we're seeing in our patients are genetic, if they're non-genetic, or if they're multifactorial. And I actually distinguish that from the question of whether they're heritable, because as we know, certain things, especially things that are caused by mosaicism, can be genetic in their cause, but not necessarily heritable if the genetic change isn't in the germline. So try to think about those two concepts when we're seeing patients with limb malformations to really help us understand the recurrence risk and provide the best recurrence risk assessment. Obviously, there are a lot of different genes that can cause limb malformation and different genetic mechanisms, so we want to try to understand what are the most appropriate genetic testing options. That can help us to understand what the best prenatal screening or testing options are, and also whether or not there are any treatment or prevention strategies. And this is the area that I think is obviously we're going to continue to think about as we make more discoveries about limb malformations to try to understand how can we actually prevent them from happening. And limb malformations traditionally have had a very important role in terms of serving as a model for understanding how malformations in general may occur. And so the limb has been a well-recognized model for understanding embryology and developmental mechanisms. And so I always think of limbs as being very complex themselves, but also serving as a model for even more complex structures. And so they can really shed light on mechanisms of normal and abnormal development in terms of the embryology and the cellular signaling that happen during limb development, and at the molecular and genetic level, the very complex mechanisms that have to occur for your limbs to develop normally. And so they teach us lessons about precise control of gene expression, and I think split hand foot malformation in particular is a good example that can hopefully help us learn more about genetic mechanisms that relate to incomplete penetrance and variable expression. And so I just wanted to show one slide of the limb bud, just to kind of remind everyone about the sort of the main signaling centers that are important in limb outgrowth and patterning. So remember that the limb buds develop around the 4th week of gestation, and most of your limb development is happening between 4 and 8 weeks. And remember that the limb bud is a mesenchymal structure. The cells that comprise the limb bud are mesodermal cells, but there's an ectodermal covering of the limb bud, and at the very tip of the limb bud, there's a thickening of the ectoderm called the apical ectodermal ridge, or the AER, and the AER helps to serve as a very important center for directing outgrowth and also patterning of the limb. Underneath the AER is a specialized group of cells called the progress zone proliferating cells, from which skeletal elements are produced as the limb grows outward from the body. And then on the posterior side, or the 5th finger side of your upper limb, or the 5th toe side of your lower limb, you have this specialized group of cells, the zone of polarizing activity, that sends signals across the limb to help direct the development of the axis of your limb from your thumb to your fifth finger. And so when we think about split hand foot malformation specifically, most people think that the problem arises in the central portion of the apical ectodermal ridge, that there's a problem with development of the apical ectodermal ridge in that area, or actually an early death of cells in that area that leads to lack of digit development in the central part of the hand and foot. So as a whole, we know that there are a number of genes and a number of loci that have been associated with limb malformation, both isolated and syndromic limb malformation. But we also know that many genetic and molecular causes are still unknown. And so I just, as an example, want to mention a study from 2009 that looked at 202 patients with a variety of different limb malformations requiring surgery. They looked at a number of established and candidate genes and only found a clear genetic cause in 11%, but they suspected a genetic etiology in an additional 21%. But I think that just tells us that there's still a lot we don't know about limb malformation. But I think that obviously exome and genome sequencing is likely to help unravel a lot of these mysteries. But what we do know at this point in time is that there are a number of important themes that I always try to keep in mind when I see patients with limb malformations. So the first one is just that there's locus heterogeneity. There are a lot of loci for limb phenotypes. And there's also allelic heterogeneity and pleiotropy. So the same locus, the same gene can cause isolated limb malformation in some instances, and in others it can cause syndromic limb phenotypes. And of course the limb phenotypes we see are highly variable, and there's a lot of pleiotropic manifestations that can come from one particular gene or one particular locus. Some molecular mechanisms that have been discovered for limb malformation are really more straightforward in a sense, in that they are associated with mutations in single genes, or in the advent of microarray technology, we know that some are associated with increased or decreased gene dosage related to deletions and duplications. But one thing that's become very evident is that there are very complex genetic landscapes that involve limb development that can lead to unusual molecular mechanisms. So, for example, multiple genes involved in limb development can be clustered in the same chromosomal region. And we know that limb genes in particular can be controlled by enhancers. So sequences that are on the chromosome that are located at some distance from their target genes, up to about 1.5 megabases away. And enhancers, it turns out, can reside within non-coding sequences, but they can also reside in coding regions of the genome as well. And so what this leads to is the possibility where you can have a genetic lesion that can affect enhancer function or enhancer gene interaction and lead to a limb malformation. And so this is just my very simple way of thinking about this, the schematic on the right sort of showing the target gene as being a light bulb and the enhancer as being an on-off switch, and the chromosome that connects the two as being a wire. And so we know certainly from split hand foot malformation and other conditions that we see that chromosome rearrangements, even balanced ones, can disrupt the interaction between an enhancer and its target gene. So that jagged line that I've shown is just to illustrate that if you have a chromosome rearrangement that breaks that connection between the enhancer and the gene, then the gene may be present, the enhancer may be present. There's no change in their sequence, there's no change in their dosage, but there's simply a disruption in their ability to interact with one another. We certainly also suspect or know in some cases that microdeletions or microduplications can alter the normal balance of enhancers in their target genes. And we also know or suspect that point mutations in certain enhancers can alter expression of the target genes. And the genes may be expressed, but they may not be expressed in exactly the right pattern. And so that sort of brings us back to split hand foot malformation for the rest of the talk. And so, so what I hope to show you is that there is an incredible amount of complexity of just this one malformation at the clinical level, the genetic level, and the molecular level. And really, it's a very important condition unto itself. It affects about 1 in 8,500 to 1 in 25,000 newborns. We certainly see it very commonly in our patients that we see through, for example, the Shriners Hospital in Greenville that serves much of the Southeast. So a lot of kids that we see presenting with limb deficiency have this condition. So it's very relevant to them and their families, and it also has, I think, a greater relevance because it illustrates many of these central themes about congenital limb malformation. So let's talk a lot more now about split hand foot malformation. So starting with the clinical aspect. So clinically, those of you who have seen this condition know that it is highly variable. So it is very tricky. It is very difficult to diagnose sometimes because it can be so mild. In other cases, it can be very severe. And in many cases, it can have the classic appearance. So it sort of runs the gamut in terms of what you might see in terms of the limb malformation itself. So you may see the characteristic split appearance. The most severe manifestation is called monodactyly, which means a single digital ray, which I'll show you in just a minute. And some of the milder manifestations can be syndactyly, and often you'll see syndactyly of the third and fourth fingers. But again, syndactyly is variable, and the mildest, I would say one of the mildest manifestations is just a slightly short central digit, but probably the mildest manifestation that I've seen is just an abnormal nail. And that can be just a curved nail. I've had families tell me they have an unusual nail that grows down the tip of the digit. I've seen a couple of cases of a double layered nail. So there can be very, very mild manifestations related to this. And of course, there can be no manifestations at all. So there can be incomplete penetrance. The other interesting thing is that the phenotype varies between unrelated people, which makes sense. It also varies between related individuals in the same family. And you can see large families with many generations of affected people with an incredible amount of variability within the same family. But even within the limbs of the same affected individual, you may see one of these manifestations in one limb, like monodactyly in one hand, and you may see a characteristic split appearance in the other hand, and then you may see a very slightly short toe in one foot, just as an example. So it can involve one limb, it can involve 2 limbs, 3 limbs, 4 limbs, 1 hand, 1 foot, all kinds of different patterns in the same individual. And so this is from a paper that I was fortunate enough to publish with a collaborator from Rome, Dr. Fiorella Guerrieri. And so I wanted just to show you kind of some of the typical findings in the isolated or non-syndromal form of split hand foot malformation. So you can see the split appearance of the hands in the upper panel on the left, and the split appearance of the feet in the 2 lower panels on the left. And then you can see some milder manifestations in the hands on the upper right. With just in this example, there's just a slight difference, for example, in this digit here, but these individuals are from the same family, and here you can see this very slightly underdeveloped second toe. And this is not at all unusual to see with this condition. So we know that there are isolated forms and syndromal forms. So the isolated or non-syndromal forms we tend to split into 2 different categories. Type 1 with hand and foot involvement only, and type 2 with long bone deficiency, or what we abbreviate as SHFLD, split hand foot with long bone deficiency. And if anyone noticed in that picture that I showed from Charles Eisenman, the photographer at the very beginning, that individual who had split hands also had underdevelopment of the bones in the lower leg. You could see an abnormality of the lower leg. And typically with long bone involvement, you'll see tibial deficiency. But it can involve other long bones. So we'll talk more about that specific phenotype as we move forward. But individuals with those non-syndromal types don't have any manifestations typically beyond the limbs, whereas persons with syndromal forms have manifestations that can involve lots of other areas of the body. So there are many different syndromes that have split hand foot malformation as a feature. So the most well-known is the EEC syndrome, so ectrodactyly, ectodermal dysplasia that can affect various ectodermal tissues, hair, teeth, nails, sweat glands, etc., and cleft lip and palate, so EEC syndrome. There's another one you may or may not have encountered, EEM syndrome, ectrodactyly, ectodermal dysplasia, and macular dystrophy that involves the vision. One condition that we'll talk more about, its chromosomal locus, is split hand foot malformation with sensory neural hearing loss, a specific syndromal phenotype that involves typically abnormality or malformation of the inner ear, causing hearing impairment, and that's linked to chromosome 7. And then Golt syndrome is just another example of a syndrome that's not really known for its association with split hand foot malformation, but can certainly cause this type of limb appearance. And there are many other syndromic conditions that can have this type of hand and foot malformation. So here's a figure from that same paper just illustrating some of these different characteristics of some of these conditions. So on the left-hand side, you can see this individual that has split hand foot malformation with long bone deficiency. So you can see the long bone deficiency, the appearance of the legs at birth. And you can see the variability here with one hand showing the typical split appearance and the other hand showing a monodactylous appearance. But in spite of this appearance, people with this type of malformation generally do pretty well. And in fact, I remember one patient that I saw that had monodactyly of both hands like this that was easily able to tie shoes like that. So again, people adapt to these malformations very well. This is just an example of an individual that has EEC syndrome, just illustrating the nail involvement, the dysplasia of the nails in the fingers, and then this individual has had surgery, but you can see the residual syndactyly in the feet. And then here are the hands actually of an individual who has a mutation in the porcin gene that causes Golt syndrome or focal dermal hypoplasia, and you can see some syndactyly, for example, in one of the feet. And again, you notice how one hand is affected here and one hand really doesn't show that split appearance, showing that variability, even though the same genetic change is presumably present. But of course, this is an excellent condition that has a lot of variability. So we know there's clinical complexity with this malformation. There's also a lot of genetic complexity in terms of how it can run in families and where the loci are that actually cause it. So we know that there are many sporadic cases. We know that there are lots of families that have this condition. So we know that there's Mendelian inheritance, and there are multiple loci. So there have been a number of different loci that have been identified over the years, and we can see autosomal dominant inheritance, autosomal recessive inheritance, and X-linked inheritance as well. As I've mentioned, there can be incomplete penetrance, and this malformation is pretty well known for that characteristic, that penetrance can be, in some cases, fairly low, and it can sort of throw you for a loop in terms of trying to figure out who in the family is at risk to transmit the trait. And there are other phenomena that have been described at various times. So with more molecular discovery, we found families that appear to have germline mosaicism for a dominantly inherited form and somatic mosaicism. There can be segregation distortion, which is a phenomenon where you can see increased transmission of the trait from affected males to their sons. so some unusual sex ratios in families. There appears to be the presence of complex inheritance in some cases where more than one locus is interacting to produce the phenotype. And that's certainly true in an animal model that I'll show you, but it appears to be true or potentially true in humans as well. Beyond sort of that genetic complexity, we then have complexity at the molecular level, the actual molecular abnormalities that are causing the malformation to occur. So we know there are single gene mutations, but we also know that with this particular malformation, there's a significant chance of finding a chromosome rearrangement as the cause. And chromosome rearrangements can be visible or they can be submicroscopic, but this is a good reason why we don't want to forget about chromosome analysis as an important tool, because a good high-resolution chromosome analysis can definitely help us with a situation like split hand, split foot malformation in terms of showing us a potentially balanced rearrangement that could be the primary cause of the malformation and could even run in a family through many generations. So we have to keep in mind that chromosome rearrangements are an important cause, and they can have variable pathogenetic effects ranging from altered gene dosage, as I mentioned, to altered enhancer gene interaction. So if we actually look at different categories of molecular abnormalities associated with split hand, split foot malformation, I've kind of divided them into 4 categories, and I wanted to go through the major examples in each category to kind of illustrate this complexity in a little more detail. So we'll talk about single gene mutations that have been associated. We'll talk about recurrent genomic duplications. We'll talk about balanced or unbalanced chromosome rearrangements that affect a common genomic location, but for which we still don't understand the exact mechanism by which they cause the condition. And we'll talk about other candidate loci. So this is just a table that sort of gives you an overview, kind of a snapshot of the complexity of the genetics and the molecular causes of split hand foot malformation. And I just want to point out, we'll go through a lot of these, but I just want to point out that most of the loci are named SHFM with a number. And so we have 6 well-established loci named in that way, and then we have 3 loci named for split hand foot with long bone deficiency, SHFLD 1, 2, and 3. This table is not all encompassing, but it sort of encompasses the major loci. And it gets confusing because, for example, there's a gene that's called SHFM1, which is at the SHFM1 locus. So you have to be careful when you're looking at the nomenclature for the locus versus the gene. So the first category is single gene mutations. Are there genes out there that have a mutation in them that causes this malformation? And the answer is clearly yes. And so one of the best known, if not the best known, causes of split hand foot malformation is the TP63 gene, or what used to just be called P63. That gene in particular is probably best known to clinicians, but there are a couple of other single genes that have been identified as causes of this condition. And there's another one in which there can be single gene mutations that I'll talk about in a little bit more detail later. So the TP63 gene is the gene that's responsible for SHFM4. There are also loci designated EEC, and so there's an EEC3 locus. So both of those loci are mapped to the same region, and the responsible gene is TP63. So this gene encodes a transcription factor that's involved in craniofacial development, ectodermal development, and limb development, which makes sense based on the type of phenotypic features that you see. You see craniofacial abnormalities, ectodermal dysplasia, and limb malformation. The gene is fairly complex in the sense that there's 6 different transcriptional isoforms and multiple protein domains that have important functions. And when you look at the mouse knockout of this gene, the mice have craniofacial, ectodermal anomalies, and they have absent limbs. And the reason is because their apical ectodermal ridge does not develop properly. So when this gene is not functioning properly, you get an abnormality in the AER. So mutations in this gene were originally identified in EEC syndrome some time ago, but it turned out that mutations in this gene could cause other phenotypes with split hand foot malformation that carried different names. So there were a couple of syndromic forms, one called limb mammary syndrome that can affect the development of the mammary glands and nipples, and one called adult syndrome. And adult is an acronym for the various features of that syndrome, which stands for, I believe it's acrodermato-unguo-lacrimal tooth syndrome. So it can affect a number of different areas, but you can see from the name there's a lot of overlap with EEC syndrome. But it also turns out that mutations in this gene can cause about 5 to 10% of isolated non-syndromal split hand foot malformation cases. So when we see a patient that has an isolated form, we also think about the possibility that they could have a mutation in this gene. And when we see a patient with EEC syndrome, this is by far and away the most important cause, with a mutation being present in about 90 to 95% of all the cases. It also turns out that mutations in this gene can cause disorders with ectodermal dysplasia and clefting, but no limb involvement. So depending on where the mutations are in the gene, what protein domain they affect, they can cause two different syndromes that don't have limb involvement, one called Hay-Wells syndrome and one called Rapp-Hodgkin syndrome. So there really are a variety of phenotypes just associated with that one gene. The second single gene I wanted to mention is located at the SHFM6 locus. So this gene is called Wnt10b. So this again is a transcription factor gene that's involved in mediating limb development. And mutations in this gene actually cause an autosomal recessive non-syndromal form of split hand foot malformation that can have other variable limb malformations. So here's an example of a single gene that can cause a recessive form. And so the point has been made in the literature that if you see an individual with a sporadic occurrence of split hand foot malformation in a family, oftentimes we think about that being a new dominant mutation, but we have to keep in mind that it can certainly be due to a recessive mutation as well. The last single gene I wanted to mention is a cadherin gene, CDH3. So this encodes protein P-cadherin, which is a calcium-dependent adhesion molecule that's involved in the beta-catenin signaling pathway and is expressed in the limb. And so mutations in this gene cause a syndrome I mentioned earlier, the EEM syndrome. Again, this is a recessive syndrome that has ectodermal dysplasia, limb malformation, and macular dystrophy. So patients have a fairly characteristic appearance of the retina with this pigmentation and atrophy. So if you see a patient with split hand foot malformation and retinal problems, this would be an important consideration. And it turns out that mutations in this gene also cause a phenotype without limb malformation, and that's a condition called juvenile hypotrichosis with macular dystrophy. So eye involvement, ectodermal involvement, but no limb involvement. So single gene mutations we know can be found by sequence analysis, but we know that recurrent genomic duplications have become an important theme, particularly for split hand foot malformation. And so there are two loci that are well known now for the association of recurrent duplications and limb malformation. So the one that has been known the longest is the SHFM3 locus at chromosome 10. So this locus had been mapped originally in multiple families that had split hand foot malformation. And in the families, the phenotype was generally non-syndromal, just isolated to the hands and feet. But what's interesting is that some of the individuals tend to have preaxial limb anomalies on the thumb side in particular. So you can see manifestations like a triphalangeal thumb. Once the molecular cause was identified as a duplication, it was discovered that those duplications also cause a syndromal phenotype called distal limb deficiencies with micrognathia syndrome. And we've known for a long time that this locus in the human is the same locus in mouse that causes a limb phenotype called dactylaplasia, or DAC. And so this is a picture of a DAC mouse, and you can see that the mouse has essentially a split foot malformation, very similar to the human. And here in this mouse, you can see more of a classic appearance in the hind foot and a monodactylous appearance in the forefoot. And so you can see similar degree of clinical variability in the mouse that you see in the human. And the mice are generally otherwise fine, so they just have this isolated limb malformation. So this is a naturally occurring disorder in the mouse. And so we've used this particular mouse model for a long time to try to understand what's going on with the human form of the condition that maps to chromosome 10. And so what was found essentially is that there is a gene in the mouse that at one point was called dactylin, but now is called FBXW4. And the mice with this phenotype have rearrangements in this gene. This gene is hypothesized to play a role in the maintenance of the apical ectodermal ridge during development of the limb bud, so that if you rearrange the gene and the gene isn't working properly, then you may have a premature degradation or apoptosis in the central part of the AER. What's interesting about the DAC phenotype is that it requires the interaction of two genes to express the phenotype. So you have to have an alteration in FBXW4. That's the major causative gene. And that alteration can be heterozygous or it can be homozygous. You get a more severe phenotype if it's homozygous, so it's actually a semi-dominant phenomenon. But you have to have a mutation or an alteration in that gene, and in the mouse it's a rearrangement. The problem is that you won't manifest that unless, at least if you're a mouse, you have homozygosity at a second locus for what's considered a permissive genotype. So certain mouse backgrounds, certain mouse strains will not express the DAC mutation. They require breeding into another strain to get homozygosity at this second locus called the modifier of DAC, or MDAC, in order to express the phenotype. And so with the human phenotype that maps to the same locus, this was a subject of study by a graduate student at Greenwood Genetic Center, Xavier de Molerat, who was working on this when I came to the center about 14 years ago. He was working on this under the direction of Dr. Charles Schwartz at our center. And they had samples from 7 families that had split hand foot malformation, 6 of whom had been linked to this locus. And so what Xavier did was he screened the FBXW4 gene for mutations by SSCP and sequencing and did not find any mutations. And so then he decided to look for rearrangements in this gene by Southern analysis and pulse field gels. So we used to run all kinds of pulse field gels in the lab to look for chromosome rearrangements in these cases, and he found through a lot of work evidence of rearrangements in all 7 cases with genomic duplications involving the FBXW4 gene and several other neighboring genes. And so this is just a very basic schematic of this region, but showing you that that here is the FBXW4 gene that's involved in limb development. It's very near the FGF8 gene, which is a critical gene in limb development. There's another gene upstream called BTRC that's also involved in limb development, and then one far downstream called SUFU, or suppressor of fuse, that's also involved in limb development. And so the original duplications extended from about here all the way down here. we now know that there are smaller duplications that actually only involve this gene and this gene called POL, but they seem to have an effect on the expression not only of this gene, but also on this gene. And so the bottom line is we really don't know exactly what's going on, because in the mouse it seems like this gene is the primary cause of the phenotype, but in human you don't have to have any involvement of that gene, nor has it been proven that there's a dysregulation of that gene by the duplications. But we know the duplications are a consistent finding, but we just don't really understand exactly how they cause the limb malformation yet. So they are easily diagnosable by array CGH. We know germline and somatic mosaicism appear possible, but the mechanism by which they cause limb anomalies is unclear. And we also don't know about that modifier of DAC gene in the human, or really in the mouse. No one has identified the gene yet in the mouse. And we think that if we can identify the gene in the mouse and then understand if there's a homolog in the human, that may be one of the culprits for explaining the incomplete penetrance or the variability in expression of the phenotype. So the other locus where you can have genomic duplication is the SHFLD locus on chromosome 17, SHFLD3. This was originally mapped in a single family, and it's associated with long bone involvement, usually the tibia, but we now know that it can be also associated with underdevelopment of other long bones like the radius and the femurs. And we also know that duplications at this locus can cause non-syndromal split hand foot malformation without long bone involvement, so just involvement of the hands and feet without involvement of the long bones. So these duplications were identified several years ago by array CGH, and now they've been identified in a number of additional families. We published the first families along with a group from Canada, 3 different families, and now there have been numerous families that have been published in the literature. The smallest duplications involve only one gene, which is called BHL-HA9. That's a basic helix loop helix transcription factor expressed in the limbs. And so it appears that probably the culprit here is this gene, and that overexpression of this one gene causes the limb phenotype. But it's unclear as to why there can be larger duplications across this region that involve this gene but do not typically cause any limb malformation. But one of the key features of this locus is the high rate of non-penetrance. So even in the families where you have a straightforward duplication involving just the BHL-HA9 gene, there's a very low penetrance, some studies have shown that males are much more likely to be penetrant than females, or to be more severely affected. So there seems to be some sort of a sex-related phenomenon going on in terms of the penetrance of the phenotype. So the third major category of molecular cause of split hand foot malformation is chromosome rearrangements that affect certain areas, but for which we don't really have an exact understanding of what's going on. And the one that's the best known is the SHFM1 locus on chromosome 7. So this locus was found not by mapping in large families like the chromosome 10 locus was found, but literally through reports of many, many different patients that had visible deletions, translocations, or inversions of this region. And some of these are de novo, some of them are familial with autosomal dominant transmission. And in some cases, you'll see other non-limb manifestations. You can see the EEC phenotype. You can see sensory neural hearing loss with inner ear malformation, which seems to be pretty consistent finding in a lot of patients at this locus. You can see dysmorphic features, microcephaly, intellectual disability, depending on what sort of chromosome rearrangement you have. So it's been known for a long time that there are several genes in the critical region at this locus. One is called SHFM1. The other ones are called DLX5 and 6 that are in essentially a tail-to-tail sort of orientation right next to each other on the chromosome. So they have been suspected to be candidates for a long time, but for a long time, none of them was really found to be consistently interrupted by any rearrangement breakpoint, and no mutations had been identified in these genes. And so the idea was that there was a suspected position effect mechanism, that the rearrangements were affecting the expression of these genes somehow and causing the limb phenotype. Now, recently there was a homozygous mutation in the DLX5 gene that was identified in a family with kind of an unusual limb phenotype. And very recently there was a heterozygous mutation identified in DLX5 in autosomal dominant split hand foot, finally giving proof that, in fact, you can have a single gene mutation in DLX5 as a cause of this phenotype. So here's a picture of the locus, and you can see here are the DLX genes sitting next to each other. Here's this SHFM1 gene. And then far upstream is this interesting gene, which is a dynein chain gene, DNC1I1. And for many years, we've seen variable deletions through this region and various translocation and inversion breakpoints that are predominantly centromeric to these DLX genes. And so what has been identified, if you think about that picture of the on-off switch and the light bulb, is that you really have the light bulb down here, you have the on-off switches up here, two of which are enhancers that are actually embedded into coding exons of this gene. So you actually have coding exons that have enhancers that have a downstream effect on the expression of these genes in the limb and in the ear and other areas. And so these were just some of our studies, just to show you an example of some of the methods that we had to use to figure out the rearrangements in two families that we had with an autosomal dominant transmission of split hand foot and hearing loss. These are some pulse field gel experiments that we used with probes to this region to actually find that these families had rearrangements in this region. And these are fish experiments to show that one of the families turned out to have a paracentric inversion, and you can see the probe there, where you see a doubling of that probe due to it being split by the inversion. And this family actually had a pericentric inversion. But we didn't know that because we didn't have a chromosome analysis on that family. So I always go back to this family and say that this is a great example of why a chromosome analysis can save you a lot of work on the front end, because had we done a chromosome analysis in that family, we would have known that this family had a pericentric inversion. There's a lot of evidence that implicates the dysregulation of these DLX genes in the phenotype. If you look at a mouse that has a combined knockout of both genes at the same time, it recapitulates the human phenotype with craniofacial and inner ear malformation. And as I said, multiple enhancers have been discovered in the centromeric region, and some of them are in coding exons of the DNC1I1 gene. One of the enhancers has been shown to physically interact with those genes, so that it's actually been shown that the chromosome will bend over and the enhancer will physically interact. And so here again is this position effect mechanism with the idea that disrupting this interaction can cause the phenotype. Mutating now, we know mutation of a gene or deletion of these genes can cause the phenotype. And it appears also that at least deletion of the enhancer area can also cause the phenotype. There's another locus on chromosome 2 that is important to mention because it can also have rearrangements associated with it and cause split hand foot malformation and a lot of non-limb malformations or non-limb manifestations. But this locus is important because it involves the HOXD gene cluster, and HOXD is a very important group of genes involved in limb development. So the genes at the 5' end of the cluster, HOXD 10 through 13, are involved in hand and foot development. And so it turns out this is yet another locus where you can have very complex mechanisms causing a variety of limb malformations. You can have small deletions that have now been discovered by array. And deletions presumably affect the Hox genes at the 5' end of the cluster. And there are a lot of other abnormalities in this region of chromosome 2 that can lead to limb malformation. You can have mutations in HOXD13, balance translocations, microduplications that cause a form of mesomelic dysplasia, and a balanced inversion that causes a phenotype in the mouse. And just to mention, there are other chromosome rearrangements that have been associated with split hand foot malformation. There are some on chromosome 2 at another location that have been associated with holoprosencephaly. And there are rearrangements on chromosome 6 that have been associated with a phenotype called MMEP, which stands for microcephaly, microphthalmia, ectrodactyly, prognathism. And there was recently a report of a large deletion in that region that causes a different syndromal form. So there's a variety of loci. There's a lot of complexity here, and there are even other loci, and this is really category 4, that have been established or thought to be loci, but for which we really don't know anything about the cause. So there's one on the X chromosome that's been identified in one family, but no one's found a responsible gene. And then there are 2 loci that have been identified for the long bone deficiency phenotype that are on 2 different chromosomes but were found in the same family, suggesting that 2 loci or more than one locus may be important in this particular family. So from a practical standpoint, what do we do with all this information? Well, I think from the clinical standpoint, I think we want to be aware of not only the variability of the limb malformation itself, but also looking for other abnormalities like signs of ectodermal dysplasia or clefting or subtle craniofacial abnormalities or hearing loss that are going to direct us to think about a particular gene locus. We want to do a careful family history and physical exam, and we definitely want to look at the parents' hands and feet and ask about a family history of some of these other minor manifestations. From the genetic testing standpoint, I think it's important to think about doing a microarray, but also important to do a high-resolution chromosome analysis typically in these cases, and to think about certain single genes depending on what the phenotype is. And with prenatal evaluation, it's important to remember that even if you're able to identify the genetic cause and offer prenatal testing for that, it's very difficult often to predict the penetrance or the severity of the phenotype. And I think with treatment, surgery is obviously the mainstay of treatment for these patients, and for the ones with long bone involvement in the legs, unfortunately amputation often yields a better functional result for them. Therapy, the use of prosthetic devices, wheelchairs, other adaptive aids. But it is important to remember that kids and adults are very resilient. And prevention strategies is just something that I like to think about because understanding things at the molecular level can give us the opportunity to use prenatal and pre-implantation diagnosis. But one wonders if there are environmental exposures that can affect penetrance or variability, pharmacologic measures we could take, or even molecular therapeutics to control penetrance and permit or not permit expression of the phenotype even when a gene mutation or chromosome rearrangement is there. So I know I'm out of time. I just want to acknowledge all the patients and families that we see, many of whom we see at the Shriners Hospital in Greenville, Dr. Schwartz and Dr. Stevenson at our center, Dr. Gurrieri, who I mentioned, Cindy Skinner, who's our research sample coordinator, many people, a number of whom are in the room from our research and diagnostic labs that have contributed to a lot of work done at our center over the years, and of course numerous colleagues. So thank you very much.
Thank you for this great presentation. There is time for one question.
So I was wondering, you mentioned several transcription factors. Do those interact with any of the SHFM sites physically, or are there target genes known? And if so, do those target genes map to any of the other loci?
Yeah, so that's a great question, Joe, and it turns out that it's looking like there are complex interactions between a lot of these loci. So for example, if you look at the P63 locus, just as an example, that P63 interacts with the chromosome 7 locus that I talked about where the DLX genes are located. So you're exactly right. I mean, as you start to try to take all these little pieces of the puzzle and fit them together, it looks like all of these genes and loci are going to be part of this complex network. All right, thank you.